Welcome back to another Explainer. Today, we're unpacking one of the absolute greatest scientific detective stories of the 20th century. I mean, look at your hands, your hair, your unique traits. We are hunting down exactly how scientists figured out the true molecular blueprint that makes you, well, you. We're going to see how a series of brilliant compounding experiments completely flipped our understanding of biology on its head forever.
All right, let's just lay out our case file right up front. We'll start with the genetic blueprint mystery, move right into Griffith's transforming principle, break down the biochemical characterization of DNA, look at the incredibly clever Hershey-Chase experiment, and finally land on the unequivocal proof.
Section 1. The Genetic Blueprint Mystery. Let's set the scene and look at the initial confusion surrounding exactly how inheritance worked.
Our roadmap here starts back in 1928, jumps forward to the 30s and 40s, and finally culminates in 1952. It took decades of sequential clues to finally solve this puzzle. As you'll see, science is rarely just one single eureka moment, it's much more like a relay race of evidence.
Picture this, it's 1926. The search for our genetic mechanism had successfully been narrowed down to the chromosomes located inside the nucleus. But there was a massive debate still raging. The chromosomes were definitely there, but what molecule was actually doing the heavy lifting of heredity? For the longest time, the prevailing thought among experts was that the genetic material just had to be a protein.
Section 2. Griffith's transforming principle. Fast forward to 1928, and this is where the plot really starts to thicken.
A scientist named Frederick Griffith was running a series of experiments with Streptococcus pneumoniae, that's the bacteria responsible for pneumonia. He grew these on culture plates and noticed two totally distinct types. Some produced smooth, shiny colonies, which we call the S strain, because they had this protective mucus coat. Others produced rough colonies, the R strain, which totally lacked that coat. And this difference was a literal matter of life and death. Mice infected with the S strain died from pneumonia, while those infected with the R strain completely survived.
So, Griffith did something really clever. He killed the deadly S-strain bacteria using heat. And, predictably, when he injected these heat-killed S bacteria into mice, the mice lived. But look at this final step, because this is where it gets absolutely wild. When Griffith injected a mixture of the harmless heat-killed S bacteria and the harmless live R bacteria, the mice unexpectedly died. And to make it even more shocking, when he checked, he actually recovered living, deadly S bacteria from the dead mice. No way, right?
An actual physical transformation had occurred. Griffith deduced that the R strain bacteria had somehow been transformed by the heat-killed S strain. He called this the transforming principle. Basically, something transferred from the dead S bacteria allowed the live R bacteria to synthesize that deadly smooth coat. It was a groundbreaking realization that genetic material could be physically transferred, but the actual biochemical nature of this principle? Still a total mystery.
Section 3. Biochemical Characterization of DNA. So the million-dollar question is, what exactly was this mysterious transforming substance? Was it the proteins everyone suspected, or something else entirely?
Between 1933 and 1944, a team of Oswald Avery, Colin MacLeod, and Macklin McCarty took up the baton. They painstakingly worked to purify biochemicals, specifically proteins, DNA, and RNA, right from those heat-killed S cells. Their whole goal was to see exactly which specific biochemical could transform the live R cells into S cells.
And they used a brilliant process of elimination. They used specific enzymes to systematically digest different components. Digest proteins with proteases, transformation still happens. Digest RNA with RNases, transformation still happens. So it wasn't protein or RNA. But look at that final row. When they added DNase, the enzyme that specifically destroys DNA, the transformation was completely stopped dead in its tracks.
I mean, the evidence was striking. DNA alone from the S bacteria was causing the R bacteria to transform. Avery, MacLeod, and McCarty concluded that DNA is the hereditary material. Case closed, right? Actually, scratch that. The scientific community can be highly skeptical. Even with this incredibly compelling evidence, a lot of biologists were just not convinced that DNA, rather than protein, was the answer, so the case wasn't quite closed yet.
Section 4. The Hershey-Chase experiment. To finally put this massive debate to rest, we turn to the brilliant 1952 experiments of Alfred Hershey and Martha Chase.
Hershey and Chase chose a fascinating test subject for this: bacteriophages. These are basically microscopic viral machines that infect bacteria. A bacteriophage attaches to a bacterial cell and injects its genetic material inside, essentially tricking the bacteria into manufacturing more virus particles. The beauty here is that the viral coat stays on the outside. So, Hershey and Chase just needed to discover exactly what was being injected: protein or DNA.
To track these molecules, they grew some viruses in a medium containing radioactive phosphorus and others in a medium containing radioactive sulfur. This is such a genius setup, because DNA contains phosphorus but absolutely no sulfur, while proteins are the opposite. They contain sulfur but no phosphorus. This meant they had perfectly tagged the DNA of one group and perfectly tagged the proteins of the other group.
So, they let these radioactive phages attach to E. coli bacteria to kick off the infection process. Once the genetic material was injected, they literally put the mixture into an ordinary kitchen blender. The agitation forcefully knocked the empty viral coats off the outside of the bacteria. Then, they used a centrifuge, spinning the samples rapidly to separate those lighter viral empty shells from the heavier bacterial cells. Now, all they had to do was look inside the cells to see where the radioactivity ended up.
Section 5. The unequivocal proof. We have finally reached the climax of our scientific mystery, where the final evidence is revealed.
Okay, let's look at the results. For the bacteria infected with viruses carrying radioactive sulfur, the protein tag, there was zero radioactivity inside the cells. The proteins did not enter the bacteria. But for the bacteria infected by viruses with radioactive phosphorus, the DNA tag, the bacteria themselves were glowing with radioactivity. The DNA had successfully passed from the virus right into the host. The results were completely undeniable. This historic 1952 experiment unequivocally resolved the decades-long debate. It established as an absolute fact that DNA and not protein is the genetic material. From Griffith's mysterious transforming principle, right through Avery's process of elimination, to Hershey and Chase's blender experiment, the true molecular blueprint of inheritance had finally been unmasked. But, understanding this elegant detective story is really just the beginning. The realization that DNA is the hereditary material paved the way for everything we now know about biology, leading directly to monumental achievements like sequencing the entire human genome. So I'll leave you with this. How is the ability to read and even edit this precise molecular blueprint going to change the future of medicine, identity, and humanity itself? It's something to think about. Thanks for joining me on this explainer, and keep questioning the world around you.